All right, class, this is the first lecture of A&P2. We are going to be studying the special sense organs as part of finishing off our study of the nervous system. Okay, most of that was covered in the previous course, A&P1, but here in A&P2 we're going to finish off with these organs. All right, so let's get started. All the special sense organs are concentrated in the head. Okay, so these are the special senses that we talk about when we refer about special senses. Now, general sensation or somatosensory perception is what we refer to as loosely touch. Any variety of touch is general sensation. So temperature, texture, pressure, pain, and vibration. Those are all various ways that we feel stimuli from physical interaction. Okay, but these are special senses. They are concentrated in the head. We have vision, we have hearing, and equilibrium. That is our sense of balance and acceleration, gravity, rotation, etc. And then smell and taste. Okay, and I like this figure because it combines all of these special sense organs. That is the special organs that are receiving the stimuli, as well as it shows in the color coding here which general part of the brain do those senses map to as far as the primary cortices, the primary cortex, to which that signal will then be designated. Okay, for example, vision goes to the thalamus, here shown in yellow, and then it synapses here and projects back to the visual cortex, which is in the occipital lobe. Okay, in the same way, hearing again goes to the thalamus, and then it goes here to this top, the superior portion of the temporal lobe. Okay, and we're going to be going through not only the anatomy of the special sense organs, what are the names of all the little parts, as well as the pathway. How do we start from the stimulus and we get to some region in the brain that is receiving that message and interpreting it? Okay, we've talked a little bit about this previously in class. This is a very important diagram to understand, have a very basic schematic about how the whole nervous system works. Okay, we have a set of receptors here on the left side. Those receptors are detecting different types of stimuli, kind of on the outside of the body, sometimes on the inside of the body. Those stimuli are received by a receptor and then communicated via the afferent system. The afferent system is bringing information from a receptor to the CNS, the central nervous system. Okay, in the brain and the spinal cord, we integrate and coordinate that signal, make sure the synapsing is to the right next neuron, and then there is an appropriate response using the efferent system. The efferent neurons, or another way of saying that is the motor neurons, which then send an outgoing signal to a set of effectors, which in most cases are either some type of muscle, like a skeletal muscle for the somatic nervous system, but also smooth muscle and cardiac muscle, and as well as glands and adipose tissue can respond to nervous system as well. Okay, so a nice general overview. Okay, as I said before, we're going to focus here on special sense organs and their receptors. So, how are receptors classified? That is, these small cellular or protein receptors that we talk about receiving different stimuli, right? Because we can walk around in the world and we can perceive different things, light and sound and taste and chemicals and vibration and all those types of things. How are receptors classified that are receiving these things? Well, that's kind of a tricky question to answer because there's multiple ways of classifying all these receptors. Okay. I just touched on general versus special. I said that general is what we refer to as touch. Okay, and by touch, it's all the I'd say TTPPV. Okay, TTPPV: temperature, texture, pressure, pain, vibration. Okay, those are 5 varieties of touch. Those are listed here. Special senses are the ones that are, again, concentrated in the head. So location around the body, okay, is one way of classifying receptors. Okay, but also we have a structural classes. That is, these are the three general structural classifications of the cells that are receiving these different stimuli. We have free nerve endings, meaning the dendrites themselves don't have anything special associated with them. The nerve endings themselves receive the stimulus. This is true of the temperature receptors and pain receptors. This next class is an enclosed nerve ending, meaning there's some type of enclosure, it's lamellated or it's surrounded by some other tissue. The Pacinian corpuscle and the Meissner's corpuscles are of this type. And then the last one here is that this neuron, this receptor neuron, is actually associating with a special cell, okay? The specialized receptor cells, some often referred to as hair cells because of these little projections that kind of project off the top of them. It looks like hair. They're not hair, but they're named hair cells because of these little cilia or stereocilia projections. It's actually this cell that is receiving the stimulus, converting it to some signal, which then communicates just like a neuron would. It's not a neuron, but it's a special kind of cell that can communicate to the neuron, and then that neuron brings the signal to the brain or to the spinal cord. Okay, so three structural classes here. Free nerve endings, enclosed nerve endings, okay, and then these specialized receptor cells that are associated. Okay, so that's another way of classifying these receptors. The third way that we can classify receptors is by the type of stimulus, okay? And so you'll see "ceptor" at the end of each of these words. You have chemoreceptor, okay? So chemoreceptors detect what? Chemicals, okay? Chemo is for chemical. So our sense of taste and smell are by chemoreceptors. We also have chemoreceptors that detect things like oxygen or CO2 or changes in pH. So those are other receptors that are around the body. They're all classified as chemoreceptors because they're detecting a specific chemical. Thermoreceptors, okay? What do you think that detects? Temperature, okay? Thermo for temperature. And so that usually has special proteins in their membrane that can detect different cold, warm, hot, etc. Nociceptor is a fancy word for pain, okay? Nociception is the sensation of pain. It's a very important class of receptors that help us to receive stimuli about noxious stimuli. Noxious just means painful. Okay, so if we put our hand over the fire, not only will we feel hot, okay, thermoreceptor, but we'll also feel pain, okay, because we should not keep our hand there, we should pull it back. And so pain is giving the signal of, hey, something's really wrong, let's stop that, let's figure that out, let's remove our body from that pain. If we touch a needle or a knife or something that's sharp, okay, nociception. Mechanoreceptor, mechano there, mechanical means a physical interaction. So some type of pressing or pulling or twisting or stretching is some type of mechanical interaction. Okay? It's a physical touch usually. Okay? So hearing and touch, proprioceptors, blood pressure, all of those baroreceptors are a type of mechanoreceptor. And then lastly, a very special case are the photoreceptors. Photoreceptors, of course, detect light. So all of these vary according to the types of stimuli that they detect. We can detect chemicals, we can detect temperature, we can detect pain, which is a unique type of touch. Mechanoreceptors is a physical interaction, and then photo is detecting photons of light. All right, and this table kind of summarizes many of those categories that we just went through. The modality here is the type of sensation that we refer to: vision, hearing, smell, taste, touch, or somatosensation. And then interoreception is a special type of internal body positioning, okay? Interoceptors help with lots of different types of sensations, okay? Vision is mediated by rods and cones. Those are the names of the receptors that are in the retina. Okay? Hearing is by auditory hair cells, so there's a listing of all the specific names. We'll go through all of these. It's just a table that kind of brings all of this information together. Gives you the receptor, the type of receptor. Here's these chemo and mechanoreceptor, the thermoreceptors. This is the organ in which they reside. So all of our touch receptors would be in our skin. Okay, taste buds contain our sense of gustation or taste. Okay. And then the type of stimulus here, okay? Light, photons of light, food and chemicals, etc. All right, so in the skin, we have a variety of these different types of classes of receptors. We talked about the Meissner's corpuscle and the Pacinian corpuscle. Those are those encapsulated or enclosed receptors. We have free nerve endings here for pain and temperature. We have special Merkel cells, which would be a special cell type that's receiving stimuli and communicating that to the sensory neuron, as well as like a Ruffini's or a bulbous corpuscle, which is a kind of a mechanoreceptor. All right, so all of these exist in the skin and give us different types of touch, or what we call general sensation. Okay, but we're not talking about skin, we're not talking about general sensation in this chapter, we're talking about special sensation. Okay. Again, another table showing you the different ways that the different special sense organs have different classes of receptors. All right, so we're going to start off with smell, then to go to taste, then go to vision, and then to hearing. That's going to be the sequence of events here. Okay. So first, the sense of smell. Okay. And as I said previously, oftentimes smell is associated with pleasant sensation. We can smell something that's pleasant, like perfume or food or whatever, something that we like to smell. But we can't forget that smell is often an alert system to warn us against something that's happening in our environment that is bad, like something is burning, or there's a chemical nearby that is smelly, like gasoline or something else. Okay, something that's rotting, so decaying flesh, or a rotten animal, a dead animal that's rotting, that will give off an odor that we can detect and say, oh, something died around here, and so that alerts us to kind of investigate more. Okay, so we need to learn the anatomy. Okay, we're going to learn all the parts of the olfactory organs. Sorry. So olfactory organs, we have the sense of smell. It's located in the nasal cavity, has two layers inside of the upper part of the nose, the olfactory epithelium, and then something called the lamina propria, which is a type of connective tissue. So let's look at that. This is tell me, what is this? Okay, what type of section is this? Okay, it would be a left lateral sagittal section of the human face, okay, the head. Okay, this would be where the eyeball is. Here's the nose, the mouth. This is the nasal cavity right here. Here's the oral cavity, the tongue, the teeth. This would be the pharynx, the oral pharynx, nasal pharynx, and laryngopharynx. And here we have the nostril, and so inhaled air would enter up into the nose. And here in yellow, we have the olfactory receptors. Okay, this yellow, expanded yellow structure here is the olfactory bulb, which then extends back into the brain. Okay, if you remember a little bit about AP1 material, we learned about the bones that form these cavities. The nasal cavity is formed at the bottom by the palatine bone and the maxilla. Okay, so that defines the roof of the oral cavity and the floor of the nasal cavity. Palatine bone, okay, would be here, and the maxilla, it would be here. Okay, at the top here, we'd have the frontal bone, we'd have the perpendicular plate and the vomer that are forming the bony nasal septum. So there's lots of bones here that we talked about. The sphenoid would be here. Sphenoid holds the sella turcica, which is where the pituitary is. And this is the ethmoid bone. We learned about the special holes that exist here. And this is called the cribriform plate. Okay, cribriform just means full of holes, like a sieve. Like if you're making mac and cheese and you drain the pasta, the water goes through the holes. Cribriform means holy, like a sieve is full of holes. Through those holes are these receptors. Okay, the olfactory bulb sits on top of the cribriform plate, and then shooting down into those holes are all of these receptors, olfactory receptors. Okay, so there's some structures there labeled. Okay, and so of course the entry of molecules enters through the nose, sometimes through the mouth, and then retronasal. Okay, retro from the back approach. So we can smell things directly. If there's airborne volatile molecules that are around. If you're smelling a rose, the rose is giving off small little molecules that smell like the rose. Or if you smell a banana, the banana is giving off volatile chemicals all the time that smells like banana, okay? Or vanilla or whatever. Anything that we smell, there's tiny little molecules that are floating around in the air that we breathe into the nose, okay? And they have an opportunity to interact with these epithelial cells, the receptor cells. And so that's a primary way that we think about smell. But I want you to think also of the secondary way, and that is the oral route. Because oftentimes when we eat food, 80%, okay, 80% of the flavor that we detect, let's say you're eating a piece of pizza, okay, got some nice cheese pizza, maybe some pepperoni, okay, put it in the mouth, you're chewing it up. Even before you eat the pizza, you're probably smelling it, but when it goes into the mouth, you're chewing it up, and lots of chemicals are being released and volatilizing, okay? When I say volatilizing, it just means they go from like a solid into kind of a gaseous state, okay? They're breaking up, they're going into the air that is in your mouth and in your pharynx, and they have opportunity, as you're kind of chewing and breathing in and out, all of those molecules are being kind of going into your lungs, and then as you breathe out, maybe through your nose, those molecules have an opportunity to be breathed back out the retronasal route, and so the food that you chewed up makes these chemicals available to then go this route, and you actually are smelling the food that you're eating, okay? And that provides 80%, okay? The vast majority of what we think is taste is actually a sense of smell. Now, there is taste, because we're talking about taste, but it is actually much less than we think it is. And this is the reason behind when we have a cold, we have an upper respiratory infection, and all of this gets congested, and your nose is plugged because there's all kinds of swelling and inflammation in your nose, so you don't have any good airflow. When you're sick, what happens to the taste of food? Okay, things don't taste good, right? Everything is a little bit bland. This is because there's no air movement through the nose, therefore there's no opportunity for these chemicals to interact with your olfactory epithelium. Okay, so most of what you taste is only the tongue and the taste buds. And frankly, there's just not a lot of discernment or distinction that the taste buds can give us. It's generally sweet, it's generally salty, it's generally bitter, but there's not really all of the beautiful variations that the nose and the olfactory receptors can give us. All right, all of this space is covered with a very specific type of membrane, okay? It's a specific type of epithelium. Now, just as a review, we covered there's 4 types of membranes in the human body, okay, all throughout the body. Okay, we have mucous membranes, serous, cutaneous, which is the skin, and synovial. Okay, so we're here in the respiratory cavities, the oral and the nasal cavities, and those are covered with a mucous membrane, okay? Mucous membrane. So, remaining moist, okay, those surfaces remaining moist is a very important aspect of this. So we're going to talk about cells and glands that are secreting mucus, and actually the olfactory epithelium are protected by that mucus. In the winter weather, the air is very dry, there's very low moisture in the air, and so our mucous membranes tend to dry out, and we're more susceptible to infection by bacteria or viruses or whatever. Anyways, mucous membrane. All of your mouth, all of your nose, all of your throat, your airway is all mucous membrane, okay? Mucus will always be open to the outside air, okay? Serous is never available to the outside air. It's always completely enclosed, okay? Your lungs and your viscera have serous membranes, okay? And they are never exposed to the open air unless you have surgery or a trauma, like a bullet wound or whatever. Cutaneous, that's skin, that's dry. Synovial, special case, that's in your joints. All right, back to sense of smell, okay? This is right lateral view, okay? Okay, here's our olfactory bulb, the cribriform plate, and we're zooming in here to the cribriform plate. Okay, this is bony tissue. Here are those holes of the cribriform plate. And we can see in yellow are your olfactory neurons. Okay, here's the dendrites, here's the cell body, and then the axons are coming up and synapsing at these little structures that we call glomeruli. Now, we're going to see the word glomeruli or glomerulus in the urinary system because the kidneys are full of glomeruli. Okay, glomerulus in Latin or Greek, I think it's Latin, means a ball, like a bundle, like a tangle of knot, a tangle of string. So these glomeruli are where there's multiple synapses happening between these olfactory receptors that are interacting with the odorants and then communicating that and synapsing with this next set of cells called the mitral cells. Okay? And those cells actually exist in the olfactory bulb, and it brings the signal via the cranial nerve 1. Okay, cranial nerve 1. O2 touch and feel, very good, Velvet. Remember, the first O is olfactory nerve. So cranial nerve 1. These neurons are bringing that signal back into the brain. All right, so this is your olfactory epithelium, which has several cell types here. There's 3 cell types that you need to know. In yellow, we talked about the olfactory receptor cells. In red are the supporting cells. They are kind of just cell spacers. They're part of the epithelium, but they're not actively participating in the sense of smell, but they're really providing some spacers between all of your olfactory neurons. And then in blue here are your stem cells. They're basal cells or stem cells, which over time can then differentiate and replace these neurons when they die, because the lifespan of these neurons is relatively short because they're constantly exposed to all kinds of things in the environment. Okay? The air, temperature, different chemicals can interact with that cell and cause it to die. And so these basal or stem cells will then get triggered to then differentiate and become a new olfactory receptor. All right, so 3 cell types here in the olfactory epithelium. Remember epithelium, what's the main thing about epithelium is it covers a surface. So this would be the roof of the nasal cavity. It's called olfactory epithelium. Here's that thin layer of mucus. Okay, the mucus is covering all of the surface inside the nose, and actually the dendrites are loosely floating around, you could say, in the mucus. And so when you breathe in some collection of odorants, which are these tiny chemicals that enter the nose, they'll have to diffuse into the mucus and then into the dendrites to interact with the dendrites. This up here above the olfactory epithelium is the lamina propria. Okay, the lamina propria, you can think of that as just connective tissue. Okay, there's loose areolar connective tissue here, some blood vessels, and the glands. This right here in the tan would be an olfactory gland. It is producing mucus, which is then secreted down onto the surface here, keeps the surface moist. Olfactory gland. This would be the gland. Remember, what is a gland? A gland is a specialized type of epithelium that makes an ooey gooey secretion. Okay? The gland makes the secretion. The duct moves the secretion from the gland out to the free surface. Okay? Don't confuse glands with ducts. All right. So each of the subsequent slides here is going to add a little bit more detail and a little bit more detail. So it looks like it's redundant, but I'm actually kind of layering on more and more detail as I show you slightly different versions of the same sagittal section. Okay. So here we have the olfactory bulb, the olfactory neurons here at the roof of the nasal cavity. We've zoomed in. Now we start to see a little bit more detail. We can see the various colors here of the olfactory receptors, suggesting that each of those different types of receptors are detecting a different type of smell. Okay? An odorant is a specific chemical that gives a certain type of smell. Now, we know that smell, when we think of like homemade cinnamon rolls, okay, delicious. You got aspects of the carbohydrate of the dough and the flakiness there, some butter, you have the sugar, you have the cinnamon itself. There's lots and lots of flavors, hundreds of flavors that are part of what we interpret as cinnamon roll, okay, or pepperoni pizza, or whatever. So there's actually a complex symphony, a complex arrangement or combination of odorants that together we interpret as a particular type of food. And so each of these various types of olfactory receptors is detecting one of those, and if 20 of them get activated at the same time, your brain will interpret those 20 as, oh, I know what that is. That's cinnamon rolls. All right. So here is the olfactory receptor cells. They go up through the cribriform plate. They meet a synapse in these glomeruli, and they interact with these mitral cells. The mitral cells are the second neuron in the chain, and the mitral cells bring the signal via cranial nerve 1 back into the brain. Okay. And so this is showing you aspects of the brain that are receiving the signal from olfaction. One special thing that you need to know about smell, olfaction, is that it is not mediated through the thalamus. Okay? The thalamus is here. Every other sensation, touch, taste, vision, hearing, goes to the thalamus first, and then synapses and projects to another part of the cortex. Smell is unique. Smell does not go to the thalamus. It has direct connections to various parts of the brain. Okay, so it goes to the medial aspect of the temporal lobe. Parts of it go to the limbic system, including the amygdala and the hippocampus. So smell is unique. All right, just a little bit more detail here. Okay, very similar image. Okay, we've already talked about. You can see the variation here in the olfactory receptors. Okay, we're going to zoom in on these dendrites. Okay, at the tip of these dendrites, you can see actually proteins that are embedded in the membrane. It's actually these proteins that are detecting the different odorants. Okay, these little dots here are meant to represent the various chemicals, let's say, that a flower is releasing that gives you the sense of smell of a chrysanthemum or a rose or a whatever. Okay? So, point of fact here, a point of distinction that you should know is when we say receptor, we could mean the cell is a receptor, or we can mean that there's the protein as a receptor. Both of those things can be true, okay? Because we refer to receptors loosely as several things. Okay, so these are receptor cells, okay, the olfactory receptor cells, the neurons. But also, this is a protein that's embedded into the dendrites of them. Those are actually G protein coupled receptors, okay? It's actually the molecular aspect that is doing the binding to these tiny little molecules, because odorants tend to be very, very small. So this is where I want you to focus your attention here on these protein receptors, and for each one of these versions of olfactory receptor cells, they express a single version of these G protein coupled receptors, and it binds to only one particular class of chemicals. Okay, so let's zoom in a little bit more. Okay, it's very similar, same. We're talking about these receptors. Now we're going to zoom in on the plasma membrane here. We can see here is the G protein coupled receptor. It's also called a olfactory receptor, okay? And so that's going to bind the odorant and then trigger the G protein to get activated, create cyclic AMP. Cyclic AMP triggers the opening of these calcium and chloride ion channels, causing a depolarization event. Okay? If you remember that wording depolarization, that's that scary stuff we talked about, action potentials. So this cell will get activated, it'll get depolarized, because the G protein coupled receptor is binding the odorant, and that sets off this chain of events, the set of dominoes that falls over, which causes depolarization. And now we're into the common lingo of the brain, which is a graded potential or an action potential. So this receptor cell gets activated, it will depolarize and cause a graded potential and then an action potential which will send the message to the brain. So all of these receptors that we'll talk about, smell, taste, vision, hearing, they are unique in the types of stimuli that they receive, but they have a commonality that when the signal is received and it's going towards the brain, every single one of those signals is an action potential. Okay? The axon is carrying the same type of thing, an action potential. And it's the brain's job to distinguish that neuron connects this structure with this part of the cortex. Okay, zooming in a little bit more, here's that G protein coupled receptor. Okay, these are the G proteins, the alpha, beta, and the gamma subunits. Gets activated, we generate cyclic AMP. Cyclic AMP binds to these ion channels, which then open up, causing a change in polarity. Whether that's depolarization or hyperpolarization, it varies from pathway to pathway, but all we have to know is that there's a change in the potential across the membrane, and that will set off the graded potential that will initiate this olfaction pathway. Okay. Second messenger in this case is cyclic AMP. Okay. Second messenger. All right, we talked about the olfactory receptor cells, we talked about the supporting cells, and we talked about the basal or the stem cells. These are the three types of cells that you find in the olfactory epithelium. Okay, we talked about the lamina propria. That's that connective tissue that is above, has some loose areolar connective tissue, some blood vessels, some nerves, and the olfactory glands that are providing mucus. Okay, so we've covered this already. Going to skip over this. Again, very similar. Here's the olfactory receptor cells. Here's the supporting cells in pink. Here's these basal cells or the stem cells. Here's the lamina propria. Okay, and then here are the holes in the cribriform plate. All right. We talked about this. Olfaction is unique because it doesn't go through the thalamus. Okay? Arriving information reaches information centers without first synapsing in the thalamus. That's what this red font is for. Okay? The ultimate destination in the brain, as I said, is hard to understand. There's no specific location that I could tell you to memorize. Where does olfaction go? Okay? Vision, very specific. Hearing, very specific. Taste is very specific. Smell, it goes a couple places, and therefore I'm not going to have you memorize those specific places. In another course, maybe in grad school, med school, they're sometimes referred to the olfactory cortex. The hypothalamus and portions of the limbic system are all receiving those senses, those action potentials from the olfactory neurons. So olfactory discrimination, that is, how do we discriminate between something that's pleasant smelling versus unpleasant smelling? Okay, all the types of foods and drink and aromas that we can distinguish. Okay, so when you think about discriminate, think about distinguish. Okay, just say this, not that. That's what that word means. Really, it is interpreting the pattern of receptor activity, okay? So just remember that when we think of one smell, probably hundreds of odorants or even tens of odorants that in combination you're interpreting as one thing, but it's actually many things altogether. All right, so this is be a close-up look at one of those olfactory neurons. Here is the stimulus, triggering the dendrites, and we could measure, okay, how frequently is the action potential sending on that neuron. We could measure this of action potentials. Each one of these would be one action potential, okay, sending it down. If we had a very strong smell, we'd have frequent, high frequency action potentials. If this is a very weak smell, we might have a lower frequency of action potentials. Okay. So as I said before, most odorants are binding to a G protein coupled receptor. We've already talked about this. That would then generate the second messenger, which we call cyclic AMP. Cyclic AMP will bind to a ion channel, which will open and allow for depolarization. All right, now I'm going to skip over the next couple slides because I'm going to talk about it in class. All right, so that was smell, olfaction. Now let's talk about taste. Okay, taste and smell, very similar because they're both chemoreceptors. Okay, so taste is also referred to as gustation. Okay, gustatory or gustation, that is what we'll refer to as taste. Okay, provides information about the foods and liquids consumed in the oral cavity, primarily on the tongue, although not exclusively on the tongue. Some of them are lining the pharynx, but we have taste receptors. Okay, many of them, again, are these G protein coupled receptors. They're just different types of G protein coupled receptors. Taste buds are the structures that we're going to examine that have all of these cells. All right, so taste buds are microscopic. That is, we cannot see them with the naked eye. What we see when we stick out our tongue, we look at that in the bathroom mirror, and we see these various bumps that are on the surface of the tongue. Those are not taste buds, those are papillae. Okay? These are papillae. There's 4 principal types, and they are distinguished by their general shape and size. The filiform, I think, looks like a flame, so filiform, flame-like. They are all across the surface of the tongue, and they give the tongue the rough sensation of or feel. Okay? They do not contain taste buds, okay? But they do provide they do have the sense of touch. So you can feel the texture of a food, whether it's creamy or rough or sharp or whatever. All of these papillae have touch receptors, but they don't all have taste receptors. Okay? Fungiform, fungi, think about mushrooms, okay, a fungus, so it has kind of this rounded mushroom shape. We have the vallate, which has kind of this inverted V, or the V shape, that's the vallate papilla. And then the foliate is kind of just a simple round pillar-like shape there. So these are papillae, okay, different size and shape, you have different names. Here in this tan color along the sides here, here, and here, and here on the top, those tiny structures are the taste buds, okay? Papillae are big, taste buds are small. Okay, so here's a look. You can see all these different papillae. In the back you have the vallate papillae that contain umami dedicated taste buds. Again, here's the 4 types: the fungiform, the filiform, the circumvallate, or the vallate. Circumvallate has an extra ring around the outside. They're located in the very back. If we were to zoom in on those little structures, you would see the taste bud itself, okay? And that's what this is. All of this structure here is one taste bud. It is filled with, again, a couple cell types, okay? Remember, in the nose, we had 3 principal cell types. We had the receptor cells, we had supporting cells, and then we had these basal or stem cells, very similar in the taste bud, okay? So here's the taste pore. The taste pore is this opening that is exposed to saliva in the opening of the mouth, okay? So tastants would be able to bind and interact with these hair cells. These cells get activated, and here is the sensory nerve fiber, which would then get communicated and send the message back to the brain, okay? There's 3 cranial nerves that mediate the sense of taste that we'll talk about in just a little bit. So we have salty taste, we have sweet, umami, bitter, and sour. Okay, so those are the 5 principal tastes that we'll talk about. Sour and salty bind to direct ion channels, okay? But sweet, umami, and bitter have G protein coupled receptors. You can see this abbreviation here, GPCR, G protein coupled receptor. Okay? Lots and lots of those are on the body. Okay? And each of these taste cells, okay, gustation cells, has one specific type of receptor being expressed on the surface. Okay, so if this cell has this particular gustation receptor that is sensitive to salt, okay, this cell will fire if it senses salty sensation. All right. So those are the cell types: the basal cells or stem cells, the receptor cells, and then the supporting cells. They're monitored by several different cranial nerves that we'll get to in just a little bit. Okay, here's a zoomed in look. This is a histological look at a cross section through. This is a taste bud here, here's a taste bud, here's a taste bud. And you can see at the base of them are these neurons, okay, sensory neurons, which are attached to these special cells, the receptor cells. Supporting cells are interspersed, and then the special gustation cells are open to the surface of these taste hairs, and they're extending through what we call the taste pore. All right, these are the 5 principal flavors or varieties of taste that we can distinguish: sweet, salty, sour, bitter, and umami. Okay, we also have water receptors, which is not a taste, but it is a receptor that's found in the mouth. So umami is hard to describe. It's a savory. You could think about it as salty. It's not salty. It's binding MSG, so monosodium glutamate, or just glutamate is what it's binding. It gives kind of this warm, savory sensation, umami. It's characteristic of beef and chicken broths, Parmesan cheese. It is sensitive to amino acids, which glutamate is an amino acid, so that's what it is binding. So these G protein coupled receptors are called gustducins. Okay, gust for sense of taste, and ducin because it's transducing the signal. Okay, but it's just the fancier receptors that are detecting the tastins. Okay, gustatory discrimination, very similar to olfaction. That is, each of these gustation cells has a unique expressed receptor on the surface, and if a tastin binds to that receptor, that cell will get activated and communicate that down the line. Okay, and that's the way that that we distinguish between different types of tastins. Okay, there's different sensitivities across human populations. If you've heard of phenylphylcarbamide, the PTC, it's sometimes a test you do in grade school or high school. Some people can taste it, some people can't. Okay, about 70% of the population can't taste PTC, and so it's just a genetic difference across human populations. With age, sense of taste rapidly declines around the age of about 50. Okay, you just start losing some of those receptors. So, how would we set up an experiment to detect how strong a tastant is? Okay, because some foods have a very strong flavor, sometimes it's very weak. Okay, it's really, really salty, or it's just a little bit salty. We taste food and it tastes bland, like, oh, that needs some more salt, because I'm not getting enough stimulation there for the food to taste good. It's like having French fries and it just tastes like potato. It needs a little bit of salt on there to make that potatoey goodness actually taste good. So how do we detect the strength of a tastant? Well, we could attach probes here and measure the frequency of the action potentials that are coming from that gustatory cell. Okay, and we have various apparatus that you could attach here. So the amount of stimulation for the receptor cell, you can see the amount of depolarization, and then that would translate to a certain frequency of action potentials, like the brrr versus the bup, 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 bup. Okay, that frequency of action potential indicates strength of signal, okay, strength of sensation. Across these 5 different flavor types of taste, sweet, sour, bitter, etc. Okay, 2 of them, I said that sour and salty interact directly with an ion channel. okay? It binds to it and it causes a direct depolarization, okay? Salty and sour do that. On the right-hand side here, sweet, bitter, and umami, those tastins bind a G protein coupled receptor, okay? So it's a couple more steps. We still end up with the same response, it's just a different way of activation, okay? G protein coupled receptor. So here's a picture for each of the 5 subtypes. Here's salty, here's acidic, meaning sour, sweet is down here, glutamate, so that's umami, bitter. So 1, 2, 3 of them have the G protein coupled receptors. These 2 here have a direct binding to an ion channel. Now, it is a bit of an old wives' tale that there's different regions around the tongue that are associated with sweet and sour and bitter. It is not the case that sweet is detected only at the tip of the tongue, okay? That's been promulgated through some early research that was done that was faulty and has since been disproven, okay? All of the flavors are detected all over the surface of the tongue. And so here is, you can have one taste bud here, has multiple taste cells, okay? And the different colors here could indicate, well, there could be taste cells right next to each other that have bitter or salty or sweet or umami, okay? So kind of knocking out this idea that specific regions of the tongue have different flavors. That is not true. Now, one more thing about flavor. This is kind of interesting, that most of these tastes that we sense through the taste bud is different than what we think of as spicy, okay? Think about a pepper, like a habanero pepper or a jalapeno or whatever, okay? Spiciness is not necessarily a flavor, okay? And so spicy is actually detected via the trigeminal nerve, okay? Remember, cranial nerve 5, trigeminal, there's 3 branches, forehead, cheek, jaw, the other one does the muscles of the jaw. Excuse me. Trigeminal nerve is for sense of touch. Touch the forehead, touch the cheek, touch the jaw. Trigeminal nerve also services the tongue, okay? The sense of touch, so if I touch my tongue, I can feel something's touching it. That is not the same as taste, of course, right? So you can have nerves that are going to the tongue that are giving you the sense of touch. That's different than taste buds. All I want to say is that spiciness, some of the flavor of a pepper goes to the taste, and some of the quality of that spiciness is actually activating the touch receptors, which is kind of weird, but it's true. All right, that is the sense of taste. I'm going to get into the visual system here, the eyeball just a little bit, but I'm getting close on time. We're going to next go to the special sense of vision, okay? Talked about smell, talked about taste, now we're going to go into the eyeball. First, we're going to talk about structures that are outside the eyeball. Okay, we have the eyeball itself, which we'll talk about. Now we have all these structures that are around it. Those are called the accessory structures. Talk about the palpebrae. That's a fancy word for the eyelid. Everybody say palpebra or palpebrae. Palpebrae is plural, palpebra would be singular. That's just your eyelid, okay? You can move your eyelid. They have eyelashes attached to them. You have different ducts and glands that are associated with them. Okay, but the eyelid is the palpebra. It's a continuation of skin from your face onto the eyelid, and it helps to keep the eyes free of debris because vision has to be clear, so you can't have any gunk covering or coating or blocking the photons of light as they're trying to enter into the cornea. So your eyelids are continuously providing an even distribution of your tears, okay, over the surface of the cornea to keep the vision clear. Okay, the palpebral fissure is just the space between your upper eyelid and your lower eyelid. That space is called the palpebral fissure. You have the medial and lateral canthus. Those are where your eyelids meet on the lateral side and on the medial side. When you wake up in the morning, you have those little gunky crusties that are in your eyes. They're usually around the medial canthus. Okay, it's just where the eyelids touch. The hinge region, you could think. Okay, eyelashes are the hair. The tarsal glands are a special type of gland. Tarsal glands are not for tears. Tarsal glands are a special oily secretion that help your eyelids from really drying out and getting gunked up. Okay, keeps your eyelids from sticking together. Okay, so it's an oily secretion, not an aqueous. The lacrimal caruncle is an opening. Well, I'll show you the pictures. The conjunctiva we'll talk about is a covering over the inside of the eyelid and the eye itself. Okay, so here is the eye, the eyeball. We're going to talk about the cornea, the iris, the pupil, the sclera. But here is the upper eyelid, the lower eyelid. Here's the medial canthus and the lateral canthus. And there's a puncta here. The lacrimal caruncle is right here. The lacrimal caruncle and the medial canthus are right next to each other. And the corneal limbus is this darker portion that is the boundary between the iris, the cornea and the sclera. Okay, we'll talk about the lacrimal apparatus. That is the system of producing and distributing tears. Okay, lacrimal apparatus produces, distributes tears and removes them from the eye as well. So here's a nice view. We'll end on this slide before finishing up for the day. Okay, we talked about the eye, the palpebra, the canthus, and then there's two little dots here. Those are the lacrimal punctum. Okay, punctum, singular, puncta, plural. This right here is the lacrimal gland. Remember, what is a gland? It's a specialized pouch of epithelium that produces an ooey gooey secretion, okay, or liquidy. So this is a right eyeball. So if you feel the tops of your eyes, right over top of your eye, and you move just a little bit lateral and then down, right there, that's where your lacrimal glands are, okay? If they're a little bit lateral on the outsides of your eyes, above the eyeball. Those glands produce tears, and then the ducts distribute them into the underside of the eyelid. Okay, so when you produce tears, the flow is from lateral to medial. Okay, and if you have an overproduction and they're like running down your face because they're spilling out and over your eyelid. But if you're just tearing up a little bit, okay, they should drain medially into the lacrimal puncta. There's openings here, and the tears will naturally kind of wick into these lacrimal puncta and drain into the lacrimal sac, which is here. And this whole structure is called the nasolacrimal duct. Okay, so it's really a mechanism to drain tears medially into the lacrimal sac, and in the nasolacrimal duct. Guess where the nasolacrimal duct goes to? The nose, okay? Nasolacrimal is the nose tear duct. Okay? So when your nose is running because you're crying, part of that could be nasal secretions that are activated. But a lot of it are your tears that were in your eye and have now drained immediately into your nose. Okay? Those are some of your accessory structures. We also have important eye muscles here. Your superior rectus muscle and your inferior rectus muscle, your medial and lateral rectus muscles for positioning your eyeballs and moving them up and down and around. Okay, those are external to the eyeball, but they are attached to the different sides of the eyeball and help to move your eye within the eye socket. All right, I'm out of time at 53 minutes here. See you guys in class.